Hello, this is Dr. A again. Welcome back for part 2 of the digestive system. So we're going to look at basically what happens from the time it leaves the stomach all the way until the end at the anus. So we finished part 1 talking about the stomach, and the food was leaving the stomach and entering the duodenum. And then we mentioned that in that section of the duodenum, the accessory organs get to add their secretions. So we are going to talk about the accessory organs. So we're going to start with the pancreas, then we're going to cover the liver and the gallbladder, and then we'll move on to what happens through the small intestine, large intestine. Okay, so the pancreas has both an exocrine and endocrine function. The exocrine function produces pancreatic juice that aids in digestion. And the bulk of the pancreas is actually dedicated to that. The endocrine portion of the pancreas produces insulin and glucagon and helps regulate blood sugar, and that we talked about in the endocrine chapter. So the pancreas is closely associated with the small intestine because it provides some of the enzymes needed for the food to be broken down and then absorbed further down in the small intestine. The cells that produce the pancreatic juice are called the pancreatic acinar cells, and they do make up the bulk of the pancreas. The pancreatic acinar cells cluster around tiny tubes that merge to form larger tubes, and then they give rise to the pancreatic duct. The pancreatic duct and the bile duct join and empty into the small intestine at the duodenum, and it is surrounded by what we call the hepatopancreatic sphincter. Again, it's a sphincter muscle, therefore it opens and closes, right, to allow the secretions from the pancreas and the bile, which come from the gallbladder, those secretions to enter into the small intestine. So this is a representation. So right here you would have the stomach. Then you have this round portion here, this like C portion is the duodenum, and the head of the pancreas is kind of nestled here into the curve of that C, and this is the tail of the pancreas. And again, the bulk of it is these acinar cells, and as they produce their secretions can flow from the cells there down into the pancreatic duct, and then the pancreatic duct, if you look here, joins in with the bile duct, which comes from the gallbladder here, right? And there's some smooth muscle cells and all that cause it to be a sphincter to open and close. And when it's open, it can release the secretion from bile and the secretion from the pancreatic duct into the small intestine, into the intestinal lumen. So the pancreatic juice contains enzymes. Those enzymes are made to digest carbohydrates, fats, proteins, and nucleic acids. So what enzymes do in this process here, which is the process of digestion, is they take big things and broke them down into small ones. So a bigger molecule, a macromolecule, and it breaks it down into its component molecules. So all the protein digestive enzymes are released in an inactive form. We talked about that when we were talking about pepsinogen and pepsin. And the inactive form is called a zymogen granule. And these guys are activated once they reach the small intestine. So they're activated in the environment of the small intestine, not in the pancreas. The problem if these zymogen granules of enzymes would be activated while in the pancreas, the problem would be it would digest the pancreas, which is essentially what happens in pancreatitis. And so enterokinase is an enzyme that can activate trypsinogen to trypsin. Trypsinogen is the inactive form of the trypsin enzyme, and it is one of the proteins one of the enzymes that digest proteins. And by the way, an enzyme is a protein, so it's a protein that digests a protein, but whatever. Moving on. So your pancreatic enzymes include your pancreatic amylase. So amylase breaks down carbohydrates. Carbohydrates are starches, such as like your rices and your pastas and your breads. And they break them down to disaccharides. So this is not yet the simplest molecule, but it breaks it down to little 2 sugar molecules. So a disaccharide is a 2 sugar molecule. Then your pancreatic lipase. So lipase is lipid breaking down, so it breaks down triglycerides to glycerol and fatty acids. So a triglyceride molecule has one molecule of glycerol and 3 molecules of fatty acids, and so it cleaves those bonds so that these molecules can be separated. And then there are 2 different nucleases that break down nucleic acids to nucleotides. So nucleic acids are your DNA and RNA, and the nucleotides are the building blocks of DNA and RNA with A's, T's, and G's and U bases there. Trypsin, chymotrypsin, and carboxypeptidases are all enzymes that can break down proteins, and they break them down to dipeptides, which are basically a 2 amino acid molecule. All right, so how are these pancreatic secretions regulated? So both the nervous and endocrine system regulate the release of pancreatic juices. Secretin is a hormone from the duodenum. It stimulates the release of pancreatic juice with a high bicarbonate ion concentration, but very few digestive enzymes. So that's the first secretion that comes out. So why high bicarb? Well, the reason is the stomach, if you remember, secretes hydrochloric acid. So stomach juice is very acidic. So on the pH scale, if you remember, will go from 0 to 14. So 0 to 7 is acidic, 7 to 14 is alkaline, okay? Pancreatic juice has a pH somewhere of 1 and 2. That's very acidic. But for these enzymes to work, these enzymes of the pancreas to work, they need a neutral pH of 7. They don't work well at this acidic pH, okay? So then what happens is the pancreas will make a very alkaline bicarb. So bicarb is over here on the very alkaline chemical, if you will, constituent here. And so it will secrete the bicarb. So a very alkaline mixes with something that's very acidic, and it becomes a neutralized substance with a pH of 7, which is ideal for these enzymes to work. And then cholecystokinin is another hormone. It is released from the wall of the small intestine as food enters in there, and it stimulates the release of the pancreatic juice that has the enzymes. So secretin causes a high bicarb ion concentration, pancreatic juice to come out. Cholecystokinin causes the one with all the enzymes to come out. Okay, so this is a fun little matching game that you can do. Again, matching the component and what it does. Okay, the liver. So the liver is our second accessory organ, and it is just as important as the pancreas. So it is reddish brown. It is located in the upper right quadrant of the abdominal cavity, and it is the body's largest internal organ. Now, the body's largest organ by weight and or surface area is the skin, but of the internal organs inside your body, the liver is the biggest one. So it is divided into right and left lobes, and it is enclosed by a fibrous capsule to protect it. So you have your right and left lobes, right one being the biggest one, right? And each lobe is separated into hepatic lobules that consist of hepatic cells that radiate from a central vein. So these hepatic lobules have the shape of a hexagon, right? It has a stop sign shape, and I'm going to show you what it looks like. And the central vein is like a dot is in the middle of the hexagon, and then it radiates all around those. You'll see some hepatic sinusoids and all of that, and some capillaries that are in there, and the sinusoids will separate groups of hepatic cells. So the way it works is blood from the hepatic portal vein will carry blood rich in nutrients from the liver. So if you remember back in the cardiovascular chapter in the part 2 there, when we're talking about the blood vessels and the venous system, we talk about the hepatic portal vein. So it's a very special section of venous circulation that connects the intestines, not back to the heart. It connects the intestines to the liver. Okay, so everything that enters the venous circulation at your intestines can flow straight to the liver. Okay, so blood comes from this hepatic portal vein, and the blood passes from the sinusoids into the central vein of the lobule and exits via the hepatic vein. So we have the hepatic portal vein coming in and the hepatic vein leaving. Okay, and then we also have not mentioned here, but we also have the hepatic artery. So hepatic artery will be coming in also. The Kupffer cells, they are stationed around in the sinusoids and around these hepatocytes. They carry on phagocytosis. So if you need to review phagocytosis, it's in the immune system. And Kupffer cells are basically specialized as macrophages, part of the immune system, and they're there to remove bacteria and viruses and other things, debris and stuff that just doesn't belong that need to be removed. And the secretions from the hepatic cells are collected in bile, and those bile canals converge to become hepatic ducts, which will merge to form the common hepatic duct. So here is the arrangement. So if you looked into the liver, you would see a whole bunch of these guys right here. So these are little stop sign shaped, hexagonal shaped hepatic lobule. They radiate, everything radiates into a central vein, right? And there's 6 corners. And at each corner, you're going to have a section of the hepatic portal vein, bringing nutrient-rich blood that just came from the intestines to the liver. You're going to have a hepatic artery bringing oxygenated blood to this hepatic lobule, so we're going to have oxygen for all those chemical reactions and produce ATP and all of that. And you're going to have the bile canal there also connected, is represented in green, and in the middle you have the hepatic vein that's draining. So it flows from the corners to the middle, from the corners to the middle, corners to the middle, corners to the middle. And the brown area here is actually all these hepatocytes. So you see each of these little cube-looking cells, right, with a nucleus in the middle, and it's kind of this light brownish color. These are your hepatocytes. And we're going to cover what the hepatocytes do, but they're very important in processing nutrients and detoxifying stuff. When they detoxify stuff, the toxins are neutralized, but then they need to leave the body. And some of them might leave through the circulation, but a lot of it's going to leave through bile. And so it's going to secrete all the stuff that it needs to secrete into bile, and bile is going to flow towards the bile duct, which are represented here in green. And so there are bile canaliculi all over, collecting all the waste and stuff that needs to go into bile. And then here's your hepatic portal vein, and you have hepatic artery, and again, the blood flows this way into the hepatic vein. So you have, the liver is really unique for that, because every other organ that you have blood flows from the artery towards the vein, and that's it. You just have the artery coming in, the vein leaving, and that's it. But in the liver, you have the artery, hepatic artery coming in, but you have the hepatic portal vein coming in, so you have 2 ins and then 1 out, which is quite unique. And again, here is a little bit more detailed, zoomed in rendering of this trio here of the hepatic artery branch, the portal vein, and then the bile duct, and how things flow through the sinusoids. The Kupffer cells are there, the hepatocytes are there, detoxifying stuff, and making bile, and bile is flowing, and then the clean detoxified blood will enter the central vein and then leave. So think of the liver as like a giant factory. It is super important to your health, and as a matter of fact, if your liver fails, you will only survive a few days. So it is impossible to, like, if it fails completely, it's completely done. It's impossible to survive without a liver. So you can survive with it at limited capacity, but once it fails completely, you can't survive. So the liver is responsible for many metabolic activities, such as the metabolism of carbohydrates, lipids, and proteins. So all your nutrients that you absorb from your intestines and travel through the hepatic portal circulation into your liver, all of that gets processed and stuff. So your liver gets to distribute. It's like distribution central, carbohydrates into the bloodstream, maybe store up some carbohydrates, so this liver glycogen, distribute the lipids. Who needs what? Do we need to make cholesterol? Do we need some triglycerides to make phospholipids? Like, what do we need, right? And then proteins, the amino acids. What amino acids do we have? What amino acids do we need? The liver has a capacity to turn certain amino acids, the non-essential ones, to convert one into another. But the essential amino acids, for example, that we need as part of our diet, it can, grab them and then ferry them wherever they're needed. So it stores glycogen. So glycogen is the storage form of carbohydrates. It's the stored form of sugar. And glycogens, the liver glycogen, its job is to maintain your blood sugar when it's low. So if your blood sugar is low, you're hopefully have enough liver glycogen stored up, and it will cleave up that liver glycogen and release it as carbohydrate, as glucose, and maintain your blood sugar while you're fasting. It will also store up vitamin A and D, vitamin B12, and iron. So because those are much needed, and especially your fat soluble vitamins can be stored up. Iron is needed to build blood, so all of that can be stored up. If you have too much of any of those stored up in the liver, it can damage the liver. So excess iron in the liver is not good. But anyway, it's a storage organ. It will synthesize lipoproteins, phospholipids, and cholesterol. So most of the cholesterol in your body, like 70% of the cholesterol is floating around in your blood, is actually made by your liver, and it's made in response to needs of the liver. So cholesterol is the building block of cortisol, estrogen, testosterone, DHEA, progesterone, I said pregnenolone, and all of these other hormones. And if you need more of those, then it will make more cholesterol so that it can build these hormones. Phospholipids are the building blocks of all your cell membranes. So it's very important for it to synthesize those so you can always be rebuilding cells and maintaining your cells. And the lipoproteins are the proteins that transport lipid in the blood. So this is your HDL, LDL, cholesterol, and your chylomicrons and stuff like that. So, it doesn't synthesize chylomicron. It synthesizes HDL and LDL and VLDL. It's the ones that it synthesizes. It converts carbohydrates and proteins to fat. So if you have excess carbohydrates or excess protein, those can be converted to fat and stored. It can deaminate amino acids, forming urea, and to synthesize either plasma proteins and clotting factors. And it can convert some amino acids into other amino acids. So again, amino acids are the building block of proteins. And so it makes your plasma proteins such as albumin, and then all the transport proteins and beta globulins and stuff like that, alpha globulins, beta globulins and stuff. And those lipoproteins, for example, too. And clotting factors are also made of proteins, and those are needed for your blood clots to form. The liver also filters the blood, removes damaged red blood cells and foreign substances, and removes toxins. So the liver's role in digestion is to secrete bile, and bile, other than having toxins and waste and stuff on their way out of the body, has some components. We're going to look at one component that's essential to digestion. So again, if you're having problems with the livers, you're going to have problems with all the dysfunctions that have been mentioned here. Okay, so what is bile made of? So bile is a yellowish green liquid that the hepatic cells secrete. It includes water, bile salts, bile pigments like bilirubin and biliverdin, cholesterol, and electrolytes. Bilirubin and biliverdin are some of those bile pigments or breakdown products from red cells. So you have a constant turnover of red cells. You can go back and review in your blood chapter for that. And some of the components are recycled and some of them are cleared. And as hemoglobin is broken down, the heme part is broken down into bilirubin and biliverdin, and that gives the bile that yellowish green color. And the bile salts are the ones that have the digestive function. So bile salts here are essential. The gallbladder, little pear-shaped organ here, lying on the interior surface of the liver, so kind of tucked away there underneath your liver, is connected to the cystic duct, which joins the hepatic duct. So we have the cystic duct, hepatic duct. Hepatic duct is going to go and merge with the pancreatic duct to form the common bile duct that leads to the duodenum. There's a sphincter muscle that controls the release of bile from the common bile duct, and the gallbladder's job is to store the bile between meals and to reabsorb water and concentrate the bile. If you have gallstones and stuff in here, it's usually crystallized cholesterol. So maybe you have chronically high cholesterol, that's a possibility. And maybe you're chronically dehydrated, that's another possibility. And so it's too concentrated, so it crystallizes and forms gallstones, which then can block. Well, they can fill up the gallbladder, but then can also block and cause blockages in the cystic duct or common bile duct or something like that. So how is bile release regulated? So bile does not normally enter the duodenum until cholecystokinin will stimulate the gallbladder to contract. So remember, cholecystokinin is a hormone, and it would be secreted when food enters that small intestine. So the presence of food then would cause bile to be released, and that gallbladder would contract and release bile there. The hepatopancreatic sphincter will remain contracted, which means closed, unless a peristaltic wave approaches it, at which time it can relax and the squirt of bile enters the duodenum. The peristaltic wave, if you remember, is what moves food through the different sections of the alimentary canal. So it would be moving food through the duodenum. And so as a wave passes, that wave allows the opening of the hepatopancreatic sphincter, it allows it to open up, and it can squirt bile and digestive enzymes and stuff from the pancreas. So what do these bile salts do? They emulsify fats. So what the heck does emulsify fat mean? It means it takes the big droplets of fat from your stomach and makes it little bitty droplets of fat. Same way as dish detergent will do with a real greasy pan. So you put soap, detergent, and water in something that's really greasy, and what the soap does is it makes all these big grease globules into really small ones, which then can be scrubbed off the pan, and they're just out down the drain and stuff like that. So that's what emulsification is taking big droplets of fat and making small droplets of fat out of it. And so that allows then the enzymes to be able to access the triglycerides and all that and break them down. And so it allows your body to be able to absorb your fatty acids, your cholesterol, and all your fat soluble vitamins. And again, the emulsification will allow those lipases to reach and digest those fat molecules that are essential for your health. Okay, so a poll question. Which of these bile components actually helps break down fat? So all of these are all bile components, right? So which one is the one that has a digestive function of helping break down fats? All right. If the gallbladder is removed, which substance then would be harder to digest? So these are your 4 macronutrients. So select which one would be logically harder to digest. And this is the practical application of that within is somebody that has had a gallbladder removed and would have harder time digesting meals that are high in this specific components. So what do you think that is? Okay, so once the pancreas and the liver have done their thing and added their secretions, let's talk about the rest of what happens. So all the magic really of absorption and digestion happens in the small intestine. So the lengthy small intestine is about 20 feet long. It receives secretions from the pancreas and the liver, and it completes digestion of the nutrients in chyme and absorbs all the products of digestion and then transports whatever remains to the large intestine. So the small intestine is divided into three sections: the duodenum, jejunum, and ileum. The duodenum is the shortest and most fixed portion of the small intestine. The rest is mobile and lies free in the peritoneal cavity. So the duodenum, remember that C-shaped that was wrapping around the head of the pancreas? That's the duodenum. So it's, it just comes off the stomach and sees into the small intestine, and that's just its shape. That's where it's at. That's its shape. That's the duodenum. The rest of it is just kind of free form and just, moves back and forth, and it's suspended from the posterior abdominal wall by a double layered fold of peritoneum called the mesentery. So the mesentery, if you will, is kind of holding all of it like strings of a puppet, and then the small intestine is hanging from it and stuff. So the mesenteries also allow support for blood vessel, nerves, and lymphatics that serve the intestinal wall. It's all super important. So here it is. So you have your stomach, your J-shaped stomach, and then you have this C-shaped duodenum. And if you had to draw it, your pancreas would be right here, okay? And then your liver would be right here. So here's your duodenum, pretty normal C shape, and then this is the duodenal and jejunal flexure. So therefore, what comes next is the jejunum, okay? And so the jejunum is like lots and lots of folds, quite lengthy, right, hanging from this mesentery, okay? And then the last section is the ileum. So that's the last section before it joins in with the colon. It joins in with the colon here at the ileocecal junction. This is the cecum, which is basically the dead end of the colon, just the beginning, if you will, of the colon on which the appendix hangs from, right? And so the food that's now waste is going to enter here, and then it's going to go up the ascending colon. Okay, so again, another quick little filling the blanks so you can grab and drag those. So put them in order that the food will travel through. Okay, so let's talk again about these intestinal villi and microvilli. So the inner wall of the small intestine is lined with these finger-like projections called intestinal villi. These greatly increase the surface area that is available for absorption and aid also in the mixing actions. So your food would be in this area where the white is, right? And it will be being broken down and digested and stuff like that. And then the nutrients are going to cross over and get into the bloodstream. Again, the fact that you have all the this instead of just this, right, makes way more surface area, so way more absorption capacity. As I mentioned in the last video, that allows this 9 feet of small intestine to have a surface area of a tennis court, which is about 500 square feet, so quite large. Each villus consists of simple columnar epithelium with a core of connective tissue that houses the blood capillaries, the lymphatic capillary, which the lymphatic capillary, if you remember, is called a lacteal. We talked about that in the immune system, and it's called a lacteal because of the fats that enter the lacteal. That's how we absorb our fats. We absorb them through the lymphatic system, and all these fat molecules make the fluid, the lymphatic fluid here in the lacteal look white. Lacto is milk, milk is white. That's why they call lacteals. So the cells that secrete mucus in the small intestines include your goblet cells and your mucus-secreting glands that are located in the submucosa of the duodenum. Your intestinal glands at the bases of the villi will secrete large amount of watery fluid that will carry your digestive products into the villi. So again, you need water. Water is very important for digestion. The epithelial cells of the mucosa have embedded digestive enzymes on their microvilli. So remember, the surface that's exposed to the lumen in those columnar epithelial cells have these little mini finger-like projections, mini microvilli, which again further increase the surface area. The digestive enzymes that these microvilli produce, these cells produce on their microvilli, are the peptidases that break down those peptides to amino acids, the sucrase, maltase, and lactase, which break down the disaccharides to monosaccharides. So sucrase breaks down sucrose, maltase breaks down maltose, lactase breaks down lactose. If you don't have enough of the lactase, you'd be lactose intolerant, and drinking milk or eating ice cream is going to give you lots of cramping and diarrhea and lots of gas. And that's the reason, is because you don't have the enzyme to break that disaccharide down, and so you can't absorb it. You don't absorb the sugar off of that, but guess who gets to absorb it and eat it? All the bacteria in your colon, and then that's what causes all these signs and symptoms that are quite unpleasant if you are lactose intolerant. Then you have your intestinal lipase. Again, same function as the pancreatic lipase, to break it down triglycerides into fatty acids and glycerols. And then enterokinase continues to activate trypsin from trypsinogen. And so trypsin, if you remember, will digest proteins. Okay, which of these produce digestive enzymes? So answer what you remember: stomach, pancreas, small intestine, or all of these. So let's talk about absorption in the small intestine. So again, the small intestine is a major site of absorption within the alimentary canal, and this is aided by the villi, which increase the surface area. The monosaccharides are absorbed by the villi through active transport or facilitated diffusion to enter blood capillaries. So your glucose requires active transport. Fructose can actually enter through facilitated diffusion, which also makes it hit the bloodstream a little bit faster because you don't have to have the expenditure of ATP. That's the difference between facilitated diffusion and active transport. Is active transport requires this expenditure of ATP for it to happen. Facilitated diffusion, it goes high concentration to low concentration through a port and does not require any kind of ATP. So that makes fructose usually hit the bloodstream faster. Your amino acids are absorbed into the villi by active transport, so it requires, again, the expenditure of energy, and are carried away into the blood. So monosaccharides and amino acids both go from the lumen through the cell straight into the blood capillaries. The fatty acids are absorbed and transported differently than the other nutrients, so they don't go from the lumen through the lining cells, the columnar epithelial cells, into the bloodstream. They actually go into the lymphatics, and we're going to talk about that here in a second. The intestinal villi also absorb water by osmosis and electrolyte by active transport. So the fatty acid molecules will dissolve into the cell membranes of the villi. Okay, so you have your villi that are aligned with columnar epithelium. They will go and merge into and enter the columnar epithelial cells, which are your enterocytes, right? Then the endoplasmic reticulum of these cells will reconstruct the lipids. So they were into single, like fatty acids, and all, they put them all back together, and these lipids will collect in clusters that will become encased in proteins. And why would you encase these lipids in proteins? To make them water soluble, because lipids are not water soluble and proteins are. And you have to transport lipids through lymphatic fluid and then through blood, both of which are water-based solutions, right? So that makes these lipid molecules in form of chylomicrons. So the lipids on the middle, in the core, and the proteins on the outside, that makes them water soluble and able to be transported then and float away through the bloodstream and the lymphatic fluid. So the chylomicrons are carried away in lymphatic lacteals until they eventually join the bloodstream. Because you remember, your lymphatic system, all your lymphatic vessels, their point is to return all that fluid back to the venous circulation. So the fats do eventually enter the blood. And so your chylomicrons will transport nothing but your dietary fats, and they'll transport them to the muscle and adipose tissue, and then also to the liver. Your VLDL molecules are made by the liver, so it's one of the lipoproteins made by the liver. They will transport your triglycerides that have been synthesized from all your excess carbohydrates. So Remember I said carbohydrates and excess carbohydrates and proteins can be made into fat. So the liver will do that, but then it has to send the fats to be stored into the fat cells. And the transport protein for those fats that came from excess carbs is VLDL. So VLDL will transport all this fat to your adipose cells and make your fat cells swell up with fats. Lipoprotein lipase unloads those triglycerides, leaving what we call LDL that have mostly cholesterol. The liver can also make LDL just with, the thing is, VLDL will carry excess triglycerides that are made from carbs. LDL mostly will transport cholesterol. And it is transporting cholesterol from the liver to the peripheral cells, to the cells of the body, because the cells of the body need cholesterol for all kinds of stuff. And so those cells of the body will take up the cholesterol from LDL via endocytosis, so it'll just engulf it and take it into the cell. And then HDL is the third lipoprotein, and what it does is it removes used up cholesterol from the tissue cells, and it returns it to the liver so it can be secreted into bile and or be used to make bile salts so that it can be, put into the digestive system and excreted from the body. So LDL, if you will, is reverse cholesterol transport. So LDL sends cholesterol from the liver to the body cells. HDL sends cholesterol from the body cells back to the liver so that it can be disposed of. So again, match the molecule with its absorption method. So it's a little matching activity. And next, so why are chylomicrons, VLDL, LDL, and HDL surrounded by protein? So it's an answer I've already given, as I was explaining, and it's a pretty simple answer. Don't overthink it. So why do these fats need to be encased in protein? So the small intestine will carry on segmentation in peristaltic waves. So there's sections of the small intestine and stuff happens in each section, but then the peristaltic wave just moves the food from one section to the next. Movements are slow. They take 3 to 10 hours to travel its length. So food spends a lot of time in the small intestine being broken down and digested and absorbed. The ileocecal sphincter is at the junction of the small and large intestine. Usually is closed unless gastroileal reflex is elicited after a meal. So meaning there's food coming, and so if there's food that's there in that section of the small intestine, is going to move it on into the colon. A strong peristaltic wave moves material through too quickly can cause diarrhea. So excessive peristaltic waves or strong peristaltic waves cause diarrhea. It can also feel like cramping and stuff for you. So then we are arriving into the largest intestine. So everything has been absorbed. Everything that's useful has been absorbed. And, all right, sorry for the interruption. My son walked in with his German Shepherd, so it's always lots of excitement when he does that. Okay, so back to the large intestine. So the large intestine absorbs water and electrolytes, and then, of course, forms and stores feces. So it is not made to absorb anything, really, and it would rather not absorb water and electrolyte there if it didn't have to, but if you are dehydrated or if you need extra electrolytes or something like that, it can absorb it from there. It is so named because simply it is has a larger diameter than the diameter of the small intestine, so it's just wider, right? It is about 1.5 meters long, so 1.5 yards, right? The large intestine consists of the cecum, which is a pouch at the beginning of the large intestine with the appendix that hangs and projects downward from it. Then the colon, the rectum, and the anal canal. But then the colon can be broken down into ascending, that goes up, transverse across, descending, and then sigmoid, which is S-shaped, which then connects it into the rectum, and then the anal canal. The anal canal will open to the outside as the anus, and it is guarded by both an involuntary internal sphincter and a voluntary external anal sphincter muscle, very similar to what we're going to learn in the urinary system for urine leaving. And so the involuntary internal anal sphincter, if your rectum is full and stuff and you need to go poop, that's going to try to start opening up a little bit and send signals to your brain that you need to go poop. And then you hopefully go find a potty and sit down. And when you're ready to poop, then you have voluntary control over that external anal sphincter, and then you actually go poop in a potty. So learning to control that, of course, is the whole point of potty training our children and stuff, so they can learn to recognize these signals. So again, here's your small intestine, and it joins here at the ileocecal sphincter, dumps all the stuff that wasn't used up, wasn't absorbed here. Here's the appendix. Part of the immune system is the appendix, we think, anyway, now. So this is the cecum, so it's like, if you will, the dead end of it, in a way. And then food is going to move, again, ascending transverse, descending sigmoid, and then rectum, anal canal, anus. It's got several things. So this is, here's a flexure. This is when it bends and folds and stuff. Same layers. This projection here is a tinea coli, and it makes these pouches called hostras right here. Okay. And it is also got some mesentery here that anchors it. All right, so the large intestinal wall has the same 4 layers found in all the other areas of the alimentary canal. Fibers of longitudinal muscles are arranged in tinea coli that extend the entire length of the colon, creating a series of pouches that are called hostras, right? We just looked at that in the picture. The large intestine does not digest or absorb nutrients, but it does secrete mucus. It will absorb electrolytes and water if needed. The large intestine contains important bacteria called the intestinal flora, microbiota, microbial flora. It has all kinds of different names, but the microbiome can refer to what's intestinal microbiome is really the correct term. So microbiome is all of the bacteria on and in your body. Intestinal microbiome is the ones that are in your intestine, large and small intestine, especially large intestine. And these guys synthesize stuff that's really, really interesting and really important for us. So vitamin K, B12, thiamine, riboflavin, which are all vitamins that you need. Also, it uses up some of the cellulose from the plants that you've eaten for raw materials. And the feces that is made is composed of any undigested material, water, electrolytes, mucus, shed intestinal cells, and the bacteria that are your intestinal flora. Both the color of feces and its odor is due to the action of bacteria. And the brown color is really from the biliverdin and bilirubin from the bowel. As it's being broken down by the bacteria, it turns the poop brown. If you have a blockage and you're not making bile, your poop will be clay colored, which is really weird. Anyway, so another, I will give you a little tidbit that's kind of fun. So if you ever look into, so vitamin K is needed for clotting, okay? And you have enough E. coli. E. coli makes vitamin K in your intestinal flora to make plenty of vitamin K as you need it, okay? It takes about a week after a child is born for it to develop enough intestinal flora, enough of the E. coli and all of that to make adequate amount of vitamin K, which is why in the Bible you had to wait until the child was 8 days old in order to be circumcised, because if they did it before that, there would be massive bleeding problems. nutrients. And it still holds true today. If they want to do it before that, they would then have to just give vitamin K injections. A lot of pediatricians have just gone to just waiting till the child is at least a week old. So here's your fun tidbit. And again, so give me a benefit of the intestinal microbiota. And then the last slide is on nutrition and nutrients. So nutrients are required in large amounts are called macronutrients. And that's what we think of when we refer to your carbohydrates, lipids, and your proteins and nucleic acids. And their energy value is expressed usually in calories or kilocalories. And those would be what you would find on the side of a nutrition panel on any kind of food that's manufactured. It will list all of that. Your vitamins and minerals are required in small amounts, so they're called micronutrients, M-I-C-R-O, micro, small, right? And essential nutrients are those that cannot be synthesized by human cells and must be provided by your diet. An adequate diet will provide sufficient energy as well as adequate supplies of your essential nutrients and your micronutrients, the vitamins and minerals and all that, to support growth, repair, and maintenance of tissues. A BMI, which is the metabolic index there, is used to determine whether a person is of adequate weight, overweight, or obese. And it's not a perfect measurement, but it kind of gives you an approximation. Malnutrition is poor nutrition that results either from a lack of essential nutrients or a failure to utilize them. And malnutrition could result from undernutrition, not eating enough. So we can think of poverty and stuff, and kids maybe not having enough meals, or overnutrition, where you're over consuming your carbs and fats and even proteins and stuff and not getting enough of the micronutrients because maybe everything that you eat is been processed and it's junk food and stuff like that. And so this is the overnutrition. It's like we're overfed and undernourished. So we get too many of these macronutrients and stuff and not enough of these micronutrients and essentials and stuff. So diet composition is super important. And the last question is, do you have any questions? And that's it. Thank you for your attention.